And so then the next challenge was, well, can we make this more sophisticated? If you remember when I showed you the slide comparing what normal grafts today look like compared to what skin looks like, there's a lot of differences. Vasculature is a really important one. And when we were doing the study, there were some examples of showing how to incorporate vasculature in skin. But if you take a close look, these are generally based on patterning channels, patterning lumen, and then seeding them with the right cells. Now, this would be great for building efficacy models that require perfusion, because you can connect these channels, you can perfuse fluid through it. But I don't think this is anywhere representative of what a microvasculature looks like in skin, or for that matter, in any other organ or tissue. So we decided not to do patterning, but to use the power of biology, which is self-assembly. And so the question we asked was, could we just print endothelial cells by themselves and let them do their thing? And we would provide them the right cues, the right microenvironment, the right niche, the right growth factors. And the way we did this was by changing what the endothelial cells see around them. So whether it's dermal fibroblasts, whether it is fibroblasts from a different source. And what these three images are showing is you can actually control the pattern of that vasculature by controlling what the endothelial cells see around them, right? And we actually did a very, very quantitative assessment of what the branching length or the persistence length, if you may. And then the data in three different colors on the graph in the top left, what they're showing is when you have different types of cells around these endothelial cells, they can instruct those endothelial cells to form very different types of networks, which would have different branching lengths. Likewise, the density of those cells would vary. So that means that the lumen diameter, the thickness of those lumen can also vary. So rather than patterning, we figured that while we can print these cells, why not provide them the right cues that they need? And then the panel of figures in the bottom shows, based on the selection of where those fibroblasts come from, what genotype, phenotype they come from, they can substantially influence the formation of this endothelial network in the skin. This is, I don't know if the video is playing on the screen, but what we were interested, we spent about a year and a half. Every time we would print the endothelial cells, they would just settle down, collapse, and form a planar layer rather than forming vascular networks. So there were a lot of tweaks that we had to do with changing the media composition, by changing the conditions that these cells see, right? But eventually what we were able to do is show that these endothelial cells, given the right conditions, actually form luminal structures. They actually form tubules. And at the bottom right, we are showing a digitally cut section of one of those blood vessels to show that there is actually a lumen present. This is a perfusible vessel. And we wanted this to be not just planar, but in 3 dimensions. And so this video is showing that these vessels actually extend between the bottom dermis all the way to the top epidermis. They are actually in 3D and not in a planar sense, right?